For the photosynthesis, I'm going to introduce certain terms. Cameras on. Harrison Nivedita, cameras on. So I'm going to reintroduce the terms we did last year. Remember the autotroph and heterotroph? There's lots and lots of content on photosynthesis. There's like new parts in the study design this year. So you might need to use these terms. That's why I'm just redoing this part, right? So autotrophs or heterotrophs, they need food, right? So remember my food example last year? So the food, if you need to make a cake, as an example, cake is your final food. You can go to the basic ingredients, whatever butter, and then make your cake. Or you can go to the shop and buy a cake and then rearrange your cake into whatever the shape you want, right? So don't write the cake example, but at the end you have to have a cake, right? So all the organisms need to have food. So all the organisms must have food. So to get the food, you can just go into the basic ingredients, or you can purchase food already made by the autotrophs, right? So autotrophs can make their own food out of the basic ingredients. Heterotrophs just rely on the food made by the autotrophs. Right, so the final food is the organic compounds. So when it comes to the autotrophs, example is plants. Right, I did exactly the same thing, that's why I'm not explaining this. So when you come to the plants, they can make the organic compounds like glucose. But to get the carbon source for this organic compound, they use inorganic compounds. So can you highlight the carbon? So this is the carbon dioxide giving the carbon for the glucose. But glucose is an organic compound. So the autotrophs like plants, they're using the basic ingredients like the inorganic compounds to make their organic compounds. But the heterotrophs are going to use the organic compounds produced by the autotrophs. They're going to rearrange the organic compounds into whatever the shape they need. Right, so these are the definitions of the autotrophs. Autotrophs can make their own organic compounds using inorganic compounds, but heterotrophs, they have to use the existing organic compounds and rearrange the existing ones into whatever the shape they need. Right, so if in case if you have to use these two terms, this is what the autotrophs and heterotrophs mean. Right? Ruhani, can't see you. Methuli. Akshara. Okay, so this is just a brief introduction. Huh? If you see in case, these are the two organisms. So the heterotrophs, example animals, including humans, we are heterotrophs. Right, you can't produce your own food, we have to rely on the pre-made food. Okay, let's go back to the coenzymes. Write down the topic coenzymes. So I introduced the coenzymes in enzymes, under the enzymes. So coenzymes can transport, I said, high energy protons, right? So they are like the buses, they are going to carry the high energy protons from one place to the other. Write down the coenzyme that we're going to use in photosynthesis. You have to change the color of the high energy proton, right? There's two high energy protons we're using in the cellular respiration. Write them down. So the photosynthesis, the coenzyme is NADP and H. So this is the loaded coenzyme because it contains the high energy proton. So can you write down on the top the loaded coenzyme? So this is the loaded one. We have the high energy proton inside the NADP. NADP at the moment carrying the high energy proton. So can you write down the cellular respiration, the remaining two coenzymes? So all of these ones are carrying the high energy proton, so they are loaded coenzymes, right? But once they go to their destination, they can unload their coenzymes, right? Can you write down the unloaded coenzyme? So if you unload the coenzyme, you can write down the high energy proton separately. There's two high energy protons here. And then the NADP. Oops, I put the here. There's no P here, right? Cellular respiration doesn't have a P. So these are the unloaded coenzymes. Can you write down at the top, these are the unloaded coenzymes? You don't have to know the full names of coenzymes, no. Right? Nicotinamide adenine diphosphate, I think. Right? No need. Right? You can just write down NADPH, NADH, FADH2. You do not need to know the term, right? But write it down, cellular respiration. So the loaded and unloaded. So can you highlight the loaded coenzyme? So when it comes to NADPH, this is the loaded ones, yeah? And unloaded means only this one, because the high energy protein is separate now, yeah? So I'm highlighting all the loaded ones and unloaded ones. So if I say unloaded version, that means you're just excluding the high energy proton. So can you identify, write down this question. If I give you the loaded coenzymes and unloaded coenzymes, which one are going to have the higher amount of energy? Right? So this is the loaded side, right? And then this is the unloaded version. So the loaded one or the unloaded one, it's going to carry a higher amount of energy. Don't think too much, it's a very easy basic question. These are exam questions. Huh? Which side will have higher energy? Loaded or unloaded? Shubham. Loaded, right? Exactly, it's loaded. So write it down, the loaded side will have higher amount of energy, right? Because it's just unloaded one doesn't have the high energy, right? So only this part gonna have higher energy compared to the unloaded one. These are like the empty buses, there's no passengers inside. So these are exam questions. All the loaded coenzymes gonna have higher amount of energy than their unloaded versions, right? Okay, so we're going to start the photosynthesis, but if you see any high energy proton in photosynthesis, right? So I'm going to write down high energy protons in photosynthesis. If you see something like this, it needs to be grabbed by the unloaded coenzyme. I normally add the plus here. If you want, add the plus, otherwise just leave it without it. Right? So the only thing you have to remember is if you see high energy protons in photosynthesis, you are adding the unloaded NADP. Right? So we will do the cellular respiration after a few weeks. Then if you see high energy protons in cellular respiration, you're adding either one of these, right? So I'm going to use this today. So remember what you have to add. But when the unloaded version combined with the high energy proton, it's going to make this loaded coenzyme. Is this clear? Nivedita, you got this? Methuli? Tanidu, can't see. Okay, so let's go back to the coenzymes and I'm going to compare it with the ATP. So I think I used the same example last year. The money example, right? So the only usable money type in Australia is the Australian dollars. But there might be people with other types of the money. Example, there might be people carrying US dollars. There might be people with Singapore dollars and whatever the other dollar, Hong Kong dollars. But all of these ones, just represent, they all going to have a value. But only thing is, if in case if they want to use the either one of the other type of money, they need to do the conversion first. Right? So if anyone needs to use US dollars in Australia, they must just convert it into Australian dollars. So same as the Singapore dollars or the Hong Kong dollars, they need to convert them into Australian dollars because that is the usable money type in Australia. So likewise, when it comes to the usable energy, there's lots and lots of things that can carry the energy from one place to the other. But only usable energy is ATP. But there are other types of things that can carry the energy from one place to the other. In photosynthesis, we are carrying the energy using NADPH. In cellular respiration, it's NADH. In cellular respiration, again, we are using FADH2. Just remember this H2, right? So all of these ones are types of energies. They can carry energy from one place to the other, but only usable energy in the cell is ATP. If in case, if you want to use the energy stored in these kind of other sources of energies, you have to do the conversion first, right? If you see an NADPH at one point, it needs to convert the energy into ATP. NADH needs to convert the energy into ATP, so same as the FADH2. What is the place where energy conversion takes place? We learned it under cellular respiration last year. If you want to do this, the energy conversion, if you have to, all these things, I said they are buses, right? So if they're the buses, they're carrying the loaded high energy proton until they come to a particular destination, is the mitochondrion, remember, electron transport chain. Right, so the conversion takes place inside the something called a reaction called electron transport chain, right, or system. So wherever you're going to see these kind of other type of sources, at one point must undergo the electron transport chain to convert their energy into ATP. So I will ask a question from this later, but remember this. Right, so let's do the ATP, the usable energy is the ATP. So you have to know the structure of the ATP. This is not chemistry, but I want you to remember at least a little bit, right? Is this slide finished? Can I go to the next one? Gopika, finish? Okay, write down the topic ATP. Write down what it stands for. We did exactly the same thing last year. Adenosine triphosphate. So there's going to be tri means 3. But what's going to be 3? Going to be 3 phosphates, right? You know this. But if I ask you to draw the ATP, you're going to draw a ribonucleotide with adenine as the nitrogenous base. Can you draw it? I said ribonucleotide, but there's going to be a difference. So include the difference and then draw the ATP. Ribonucleotide, right? So you have the pentose sugar, but it's ribonucleotide, so the sugar needs to be ribose. And adenine is the nitrogenous base, and then this is the phosphate, right? So this is the ribonucleotide with the adenine as the nitrogenous base. But the difference is that not only one phosphate, there has to be 3 phosphates because adenosine triphosphate. So we are drawing 2 more phosphates. So this is what the structure is. I need you to remember at least a little bit. When we do the trial exam, there might be questions asking the structure. Right? If you know the ribonucleotide, just add 2 more phosphates. So if I run or if I walk, I need the energy. So I need to spend the energy packets, right? ATP packets. So we're going to get the energy by breaking down the last phosphate bond. So if you run, you're using the ATP packets by breaking down the last phosphate bond and releasing the energy stored inside the ATP. Right? So this is the bond that's going to be broken down. So draw the ATP after breaking down the last phosphate bond. Because we are breaking down the last phosphate bond, now ATP has only 2 remaining phosphates. Ribose R. But the broken down phosphate you must write as the inorganic phosphate. This is what you're going to write throughout the photosynthesis. Write it down. This represents inorganic phosphate. Right? So inorganic phosphate is nothing else. It is just the broken down, the phosphate, right? So this is the phosphate. But we have just separated it out because we just broke the bond to get the energy out. And write down the name of the newly formed compound. I did exactly the same thing. I think I did this part, I think, last year. So there's 2 remaining phosphates. So instead of saying adenosine triphosphate, you're going to say because we just change the triphosphate, we have only 2, so it becomes adenosine diphosphate. So ATP becomes ADP. But don't forget the broken down phosphate. You have to always write down your inorganic phosphate. Right? So I had ATP, I used the ATP, and for my whatever the work. So for my run, I used ATP, and ATP became ADP and inorganic phosphate. But all these ADPs will go back to the mitochondrion and put the phosphate back to the ADP. Right? So inside the body, we are not just generating ATPs from the basic ingredients. They are always going to be recycled. Because ADP can easily convert back into the ATP if you put the broken phosphate back, right? So this is what happened in the cellular respiration. We are putting the phosphates back to the ADPs, right? So the cells throughout the body are going to use the ATPs and end up with ADP and PI. But all the ADPs will go to the mitochondria and then they're going to get the phosphate back to the ADP. So as soon as the third phosphate comes in, the ADP becomes ATP. Right, so this is a cycle. We're going to call it as ATP ADP cycle. So all the ADP is going to get their phosphate back and recreate the ATP. This is exactly the same thing, but this is easy. This is the ADP ADP cycle. Is this clear? So this is the ATP ADP cycle, right? So if you're born with 100 ATP, if you run or walk or whatever, you will break down your 100 ATP into 100 ADP and PI. But all these broken down ADPs can be recycled by putting this phosphate back. So it will recreate the ATP 100 back again, right? So we do not have to just use pre-made the ATPs all the time. It can be recycled over and over and over. Right, the bond is the one that's going to break and release the energy. Nothing happened to the inorganic phosphate, right? You can recreate the bond. Okay, is this clear? So this is the ATP ADP cycle. Is this clear? Right. So this one here. So if there's only one phosphate, we're going to call it as adenosine monophosphate, right? So that's the normal nucleotide, yeah. But For this one, there's 3-phosphate, so we're going to call it as adenosine triphosphate. That's the difference between the RNA nucleotide is adenosine monophosphate. Mono means one, right? I'm answering a question. So that's going to be 3-phosphate. So instead of saying mono, you're going to change the term to tri. Next one. Is this clear? Okay, next part. Photosynthesis. Write down the balance equation. You're doing an anabolic endogonic reaction. Build up something, don't break it down, don't accidentally write the cellular respiration. Photosynthesis always builds up something. Right, so carbon dioxide plus water breaks down into, build up into glucose and oxygen, right? I will change the colors later, but if you want, you can use the same thing, because carbon dioxide becomes glucose, water becomes oxygen. Yeah, I use the same color. You have to have a balance equation. So the photo means synthesizing something using sunlight. This is 3, 4. If you don't write sunlight, you will lose the whole mark, I think. If I'm the examiner, I won't give a single mark. There's no synthesis if there's no sunlight, because this is photosynthesis. So what you're going to synthesize is the glucose, right? So to balance it, you have to have the 6s, except the C6H12O6, it's already have the 6s. So the main thing is for the photosynthesis, the first thing the question going to ask is to write down the balance equation. You must write sunlight and you have to balance it. Huh? It is not your 1 and 2, you must balance it. Whether it asks for the balance equation or not, just write down the balance equation with the sunlight. The pigments or anything, they're not compulsory, but you have to write the sunlight, because without sunlight, there's no photosynthesis. So the photosynthesis takes place inside the chloroplast. Can you draw the chloroplast? I skipped over the organelles. Huh? So this is the first time I'm doing in 3, 4 the organelle. Draw the chloroplast. How many membranes it gonna have? I need thylakoid. Don't draw microscopic ones, just do something big. Thylakoid, top of the thylakoid add the pigments. The stack of the thylakoid is the granum. The liquid is the stroma. And you have to have the outer membrane and inner membrane, because there's a bacterial origin. Finish? So there's no like foldings or anything like cristae. There are two flat membranes. Inner membrane and outer membrane. Outer membrane has a bacterial origin, remember? It came from the prokaryotic organism. Outer and inner membrane. And the liquid is the stroma. And the individuals, the disc is the thylakoid, and you're going to have a stack of thylakoids. You can just go from 3 to 7, right? So you can just end up with 3 thylakoids, or you can just, whatever the number in between 3 to 7. I have 5. So there's going to be a couple of grana. And they're connected by lamella or something, no need. And the liquid is the stroma. So you have to know where the thylakoid and stroma, right? Because there's going to be reactions taking place. On the top of the thylakoid membrane, the light capturing pigments. So the single disc is the thylakoid, and the stack of thylakoid is the granum. Right, we did every single thing last year. So you have to remember thylakoid because the light dependent reaction takes place, and the light independent is in the stroma, so you have to know the locations. Okay, so look at the diagram and then look at the photosynthesis reaction, and then write down the requirements for the photosynthesis. What are the 4 things the plants need to start the photosynthesis? So if you're a plant, what are the things that you need to start the photosynthesis? Right, anything that's going to go into the reaction are inputs and the products are outputs. Right, so what are the requirements for the photosynthesis? There are 4 things you need to have to start the photosynthesis. 4, write down all the 4 things. Right, all the inputs, right? Yeah, the red dots are light capturing pigments, huh? That's correct. So if I'm a plant, I need carbon dioxide as my carbon source, because this is the carbon that's going to go into the glucose. If I'm a plant, I need water. They are the 2 main inputs. The sunlight is not a physical thing, but just write down, there has to be sunlight present. And there has to be the light capturing pigments present. Let's do pigments in detail. And then we're going to do a question, so listen carefully. So photosynthesis is quite low. Has lots and lots of stuff added, Rubisco and stuff. C3, C4 chem plants. Right? Okay, let's do the pigments. So I'm going to go to the next slide and we're going to do the pigments separately. So we are only considering the green plants because this is the most abundant plant, right? So throughout the everywhere you can see the green plants. So if I consider the green plants, so if you have a practice, think about whether you're using the green or not, right? If the plants are not green, that means they're not green plants. If you use the green plants going to have the primary photosynthetic pigment. Is the chlorophyll A. It's the little a, don't write the capital A. Right, so the primary photosynthetic pigment present in the green plant is the chlorophyll A. Chlorophyll A is the primary photosynthetic pigment. So let's redraw the thylakoids. So if I redraw the thylakoid, I swap the colors, the previous ones. So these ones, the little dots are the chlorophyll, right? Then the pigments, but the specific pigment type is the chlorophyll A. So the pigment's function is to absorb sunlight. So initially the pigment's going to get the white light. So this is the white light coming to the chlorophyll A pigment. So white light is made out of 7 colors, right? That's the rainbow. So white means it's just the combination of 7 wavelengths. So we are not using the term color, instead we are using the term wavelength. So white light is made out of 7 colors or 7 wavelengths. But all the wavelengths just come into the chlorophyll A. Out of the 7 wavelengths, some of the wavelengths going to be fully absorbed to produce for the photosynthesis reaction. Right, there's going to be out of 7, there's 2 wavelengths going to be fully absorbed by the chlorophyll A. Some of the wavelengths little bit important, so they might be just partially absorbed. Some of the wavelengths not needed at all. The full spectrum is going to be reflected. Right. So out of the 7 wavelengths, some of these wavelengths need to be present for the the photosynthesis to take place. So the two of the wavelengths going to be fully absorbed by the chlorophyll A. Write down the two wavelengths. I did this last year. What are the two wavelengths going to be fully absorbed by the chlorophyll A? This is only for the chlorophyll A, right? So the primary photosynthetic pigment, chlorophyll A, need 100% of red and violet. So these are the two wavelengths that are going to be absorbed like 100%. Some of the wavelengths just partially absorbed, we don't need them, but the full spectrum that's going to like reflected, because it's the least important wavelengths for the chlorophyll A, is the A, everyone knows this, yeah? Is the green wavelength. So why the green plants are green? Because the full spectrum going to come into your eye and you're going to see the leaves in green color, because all the green plants, if they have chlorophyll A, the chlorophyll A gonna reflect the entire green spectrum. Do not write colors now, it's not a color, it's a wavelength. So the entire green wavelength going to be reflected by the chlorophyll A. It comes into your eye and you're going to see the leaves in green color. Right? All you have to know is the two most important photosynthetic wavelengths for the green plants. And the least important wavelength is the green. I did all these things last year, isn't it? I think this is just a repeat. Right, so if you do a prac, you might design practicals to investigate whatever the wavelength, right? So cellophane wraps and stuff. So this is only for the primary pigment. The primary pigment, photosynthetic pigment for green plant is the chlorophyll A. So these are the requirements of the chlorophyll A, right? So chlorophyll A reflects the entire green spectrum and fully absorb the entire red and violet spectrums. Right, so the full refraction coming to your eye and you're going to see it in that color, specific color. Right, so this is what happened with the primary pigment. Primary pigment is all over the place in green plants. So all you see is the reflection coming from the chlorophyll A. But other than the primary pigment, there are accessory pigments. Right on the topic, we're doing accessory pigments. But most abundant, most frequent one is the chlorophyll A, but there has to be other accessory pigments as well. Right, I'm going to go to the next page to do the accessory pigments. Done to finish? No one is writing this down, right? I can go to the next page. Okay. So I finished the green plants, the main pigment. And then we're going to do the accessory pigments. Right, so the particular wavelengths, the red and violet, the green, only for the chlorophyll A. Other pigments inside the plant going to absorb and reflect at different wavelengths. Write down the accessory pigments, 3 of them. Chlorophyll B, chlorophyll C, carotenoids. Right, so they are absorbing and reflecting at different wavelengths. I think everyone knows the carotenoids. Carotenoids reflects, you don't need to know the absorption, reflects the orange or the yellow, I think. So that's what happened in winter. Under normal circumstances, chlorophyll A is going to mask the reflection coming from the other pigments because that's the most prominent one. So if it is the most prominent abundant pigment, all you see is the reflection coming from the green wavelength or the chlorophyll A. But during winter, the chlorophyll A dies. So as soon as the chlorophyll A is gone, you start seeing the reflection coming from the other pigments. That's why it starts showing the yellow or orange in winter or autumn. Right, so as an example, why the leaf turn orange in autumn? Because you start seeing the accessory pigments reflection. But in spring, the chlorophyll A grows back, still you start seeing the green again. Right, no need the winter part, but remember it's when it comes to the photosynthesis, the main photosynthetic output coming from the chlorophyll A. But for the green plants or any plant, it's not just one pigment that's going to contribute. There's going to be other accessory pigments absorbing at different other wavelengths. So when it comes to the total photosynthetic output, it's the contribution from the primary pigment plus the accessory pigments. This is important. Huh? So the photosynthetic output or the glucose production is going to depend on the absorption coming from the primary pigment. But it is not just the primary pigment, there are contributions from the accessory pigments as well. Right, so primary pigments absorb at different wavelengths other than the accessory pigments, right? All the accessory pigments just absorbing at different wavelengths. So if they absorb at different wavelengths, it just increases the efficiency, right? It increases the production. At the end, all these pigments going to contribute to the final photosynthetic output. There are questions coming asking like this, so try to understand what I'm saying. It is not the main, not only the main primary pigment going to contribute to the final photosynthesis, the glucose production. Write down photosynthesis means you're measuring by the amount of glucose. So it's not just the primary pigment, you have to consider the contribution coming from the accessory pigments. Is this clear? So I'm going to draw two graphs. Right, so I'm going to draw two graphs. Just draw the two graphs. The absorption spectrum and the action spectrum. Right? Let's do the absorption spectrum. So let's draw the absorption spectrum. Axes, huh? Look at the axes. So the name of the spectrum is, the graph is the absorption spectrum. So wavelengths on the x-axis, different wavelengths. I don't think you need to remember the wavelengths, so no need. If you have to remember the color, but not the wavelength numbers. But at different wavelengths, what is the absorption? So x-axis is wavelength, the y-axis is, depending on the wavelength, how the absorption is going to be taking place. Right, so the graph, the title is absorption spectrum. So I'm going to now draw the absorption spectrums for individual pigments. Right, so let's start from the chlorophyll A. So chlorophyll A is going to absorb at two different wavelengths, right? So one is going to be at the, whatever the number, the red color, and the other one is the violet wavelength, right? So it's going to produce two peaks, one for the whatever the color, the other one is for the other color, right? So there's going to be two peaks. The only thing you have to remember is that there's going to be two peaks, because there's going to be higher absorptions at the two peaks, two wavelengths. Just draw it like something like this. So just go down. Right? First one and then the second peak. So label the thing line as the graph as the chlorophyll A. So the red one represents the chlorophyll A, because it's going to absorb at two different wavelengths. Right, 650 and 450. Right, so either one going to be, I don't know which one, but one is for red, the other one is for the violet, right? So there's going to be higher absorption at these two wavelengths. But it is not just the chlorophyll A that is going to contribute. There's going to be other accessory pigments. So chlorophyll B, as an example, it's going to absorb at a different wavelength. Yeah, so this is the chlorophyll B's absorption. Just draw whatever the graph, no need the shape. You don't have to memorize these graph shapes. And then carotenoid is going to absorb at a different wavelength. Right, so each color, each line represents the absorption of different wavelengths, different pigments at different wavelengths. So the absorption spectrum, you can see three different lines, right? So individual, each line represents the individual pigments absorbing at different wavelengths. So this is what happened with the individual pigments. They absorb at different wavelengths. This is clear? So because of the absorptions, the final output is the photosynthesis, or the action is the amount of glucose produced, right? But for the amount of glucose produced, we're going to get the contribution from every single pigment, right? So I included three different pigments absorbing at different wavelengths, contributing to the final glucose production. So when it comes to the glucose production, so I'm going to draw the second graph. right? So the x-axis is still the wavelength, but the y-axis is the amount of output or the photosynthesis output or the glucose production. If the pigments absorb at different wavelength, how it's going to contribute to the total glucose production, right? So we're going to name this graph as the action spectrum. Write down action means the glucose, right? Photosynthesis is the glucose production. Not to glucose, just the glucose production. Right, so this one, the glucose production is the action. So when it comes to the action spectrum, because of the absorption happening at individual wavelengths by different pigments, it's going to, every single pigment going to contribute to the final glucose output, right? So they're going to absorb at different wavelengths and contribute to the final glucose production. So for the action spectrum, you just combine everything and only going to draw one line, right? So the total glucose production. So it is the combination of every single absorptions. So as an example, look at this. So around like 400 wavelength nanometers, you can have a bigger photosynthetic output because there's going to be multiple pigments absorbing around this region. And then the glucose production will just drop around the middle range and it will just slightly go up at the end according to my graph, right? So that's what you're going to draw. There's going to be higher photosynthetic output and then it will drop and then slightly go up. But there's only one line, right? So the action spectrum means the contribution coming from individual pigments to the glucose production. Whatever the shape you drew here, just get the cumulative action and then draw one line. Definitely there's going to be stacks on photosynthesis, huh? There's CRISPR parts again, biofuel again, not again, new parts, chemistry parts. Is this clear? I'm going to send you a question. Do the question. Take a screenshot. No? That's going to be other questions as well. At the end, I will give you the question. I'm going to share the screen, but the concept coming from these two graphs. No? So the first one is the absorption spectrum. We are showing all the absorption, individual absorption by different pigments, but it's going to contribute to the total action, which is the glucose production, right? So glucose production is just one line. I'm going to share the screen, take a screenshot. No? Okay, answer the question, 3 minutes. Wait, I'm changing the question. This is the second one. Let's do something else first. Okay, write down the question. Draw the graphs. I'm going to draw an absorption spectrum and the action spectrum. Okay, so I'm drawing the absorption spectrum for the chlorophyll A, action spectrum for the chlorophyll A. So this is the chlorophyll A. And this is the action spectrum. Write down the question. Right, so the question is, if the chlorophyll A is the main primary pigment, why the chlorophyll A's absorption spectrum significantly different to its action spectrum? So why there is a difference between the chlorophyll A action and absorption spectrums? Right, so if the chlorophyll A is the main pigment, why it is not exactly the same as the action spectrum? Answer this question first, and then I share the other one. Why there's a difference between the chlorophyll A's absorption spectrum, or why chlorophyll A's absorption spectrum not exactly the same as its action spectrum? If the absorption contribute to the action, why they're not identical? Finish. Right, so the answer is, when it comes to the absorption spectrum shows the individual absorption only by the chlorophyll A, right? But when it comes to the action spectrum, it is just the cumulative action of the actions coming from every single pigment. So the action spectrum shows the actions from other accessory pigments as well. So it's not going to be identical to the chlorophyll A, right? Because this represents the actions from other pigments as well. Is this clear? So I'm going to share the next one. Let's do the next one. So this is a application question. Just do it and get the 1 mark. It's like absorption and the action spectrum on the same graph. It's the same thing, but they ask a different way. It's the same thing. My question has the answer for this. So the action spectrum is the dotted line, right? This is the absorption spectrum. Is this finished? What evidence on graph indicates that chlorophyll A is not the only pigment involved in photosynthesis? If it is the only pigment, absorption spectrum's line needs to be identical to its action spectrum, right? So the black line needs to be at the same spot as the action spectrum. But the rate of the photosynthesis is, the line is a little bit higher. That means there must be other accessory pigments contributing to the photosynthesis. It's not just the chlorophyll A, right? So there has to be other pigments absorbing at different wavelengths. As an example, if you take just the chlorophyll A, you can't have higher amount of output in this section, right? So in between 450 and like 550. Okay, next one. Write down the balance equation again. I'm going to change the colors. Remember the light dependent reaction. So we are using water, break it down and get oxygen. But for the first reaction, we are using sunlight. And the second reaction, we are using carbon dioxide, building up into glucose. Just change the colors, otherwise you're going to have to memorize this. Don't balance the equation now, no need. So the two reactions, the same two reactions, right? So light dependent and then light independent reaction, use the same two colors. So we're using the red ones for the light dependent reaction that means the water going to be broken down into oxygen. But the second reaction is the carbon dioxide building up into glucose. Don't swap them. Okay, so this is what happens. Write down the location of each reaction. It's the same thing we did last year. Right, so light dependent depends on the light. So it needs to happen somewhere which can trap the sunlight. So the location is the chloroplast thylakoid membrane. Don't write just the thylakoid membrane. I want you to write down somewhere it's part of the chloroplast. And light independent, there's no light trap. So it can happen in stroma, but this stroma is part of the chloroplast as well. Right? Can you go back and write down? Because we haven't done any organelles, can you write down whether every plant can have the chloroplast or not? Go back where we draw the chloroplast and write down whether every single plant cell can have the chloroplast or not. I did all these questions last year. Just pick the answer and write down the reason. So the chloroplast is the organelle we need for the photosynthesis. Photosynthesis is going to rely on the sunlight, synthesizing glucose by trapping the light. So the question is, can every plant cell undergo photosynthesis? I gave all these answers last year. Is this true or false? Is this true or false? It is false, right? Because root cells don't have chloroplasts. So photo means the sunlight, you're not getting sunlight into the root cells to get the roots are underground, right? So this is a very, very popular question. But the only thing is you don't have the organelles, but here you can get this question, right, under photosynthesis. So chloroplast not present in every single plant cell, it's a cell wall. Remember I did this last year? Cell wall, cellulose cell wall present in every single plant cell, but not the chloroplast. Because the root cells, they are underground, no sunlight, there's no point of having chloroplasts. Without sunlight, no synthesis. So let's do the light dependent reaction. Okay. So if I break down something, the energy going to be released, right? So energy release means it's the net amount of energy. For every reaction, you need to absorb energy, but the net amount of energy going to be released. So the energy can be in different forms, but if we break down something, energy going to be released in the form of main usable energy and in the form of high energy protons. Write down this somewhere. Right, so if I use the term energy, we're going to have the energy in the form of ATP, the most usable one, as well as the high energy protons. Right, high energy protons getting from the water. So let's split the water molecules. I'll do this part again. You need to see the H plus. Right, so go back. So light dependent, don't take anything else. We are taking only the red ones. Right, so you take water in the presence of sunlight, going to split it into oxygen. So water in the presence of sunlight, split it into oxygen. So for photolysis or splitting the water molecule, I can draw the water molecule like this. So we're going to use the sunlight to split the water molecule like this. Right? So this is photolysis because you're using the sunlight for this split. So if I use the sunlight as the energy source to split this molecule, but after splitting, you're going to get the protons out or energy out because you're breaking down something. Write down the two types of energy that we're going to get. So for this example, you're going to, because we are splitting something, the main energy I said ATP, and you can see the high energy protons. Use the coenzyme and grab the H plus. Use the unloaded coenzyme, grab the H plus and convert it into the loaded coenzyme. Right, so photosynthesis can have the unloaded coenzyme NADP. So NADP going to come and grab the high energy proton and it's going to become the loaded coenzyme NADPH. Right, so the most important thing of doing this light dependent is to get the energy out from this water molecule. So the main product of light dependent is the energy. Right, so I'm going to change it. So what you're going to end up is the ATP plus NADPH. Can you circle this? This is the main product. Because this is the energy. So why the light dependent reaction takes place is just to get the energy out. Right, so the most important thing of this water split is, right? So the most important thing for the water split is to get the energy out. Right, so this is the main product of light dependent reaction, but the remaining oxygen is going to combine with another oxygen atom and it's going to become O2. But oxygen is just a byproduct, it's not the main product. Write down what happened to the oxygen gas. Can you highlight the oxygen? So this is the oxygen atom, it's going to go into the oxygen gas. Oxygen is a gas, it needs to release from something, right? So write down what happened to the oxygen. So if your plant is contained inside like an enclosed environment, this oxygen can be used for the cellular respiration, right? But oxygen needs to be released to the outside. If the oxygen is going to go into the outside, it's going to go through the plant, the little holes, the something. Right, underneath the plant, there's going to be little holes called stomata. So stomata's main function is to let the carbon dioxide in for the light independent reaction. Because carbon dioxide is the main carbon source for the light independent to make the glucose. But at the same time, because it's just an open little hole, the oxygen can go out as well, right? So carbon dioxide can come in and oxygen can release to the outside. But the main function of the stomata is to get the carbon dioxide in, yeah. Oxygen, we don't need oxygen, it is just, plants don't need oxygen, it's just a byproduct. Plants don't undergo light dependent to get the oxygen out, it is just for the energy. Is clear? Okay, so write down the oxygen as a byproduct. The main product is the energy. So the oxygen end up in the oxygen gas. So this oxygen will go out through the stomata. Write it down, these are the things you need to know. Can you just highlight oxygen being in the oxygen gas? Write down the inputs and outputs. Anything that go into the reaction are the inputs, the products are the outputs, right? We did exactly the same thing last year. Only thing I'm adding is NADPH, right? So write it down. I need 3 inputs, 3 outputs. Start with the outputs and then go backward. So you can see the outputs, right? Just convert them into the inputs by looking at the outputs. Finish? Okay, so let's write down the outputs. If you write oxygen, you don't have to memorize these things, right? If you write oxygen, the opposite side needs to have water, because water's oxygen atom converted into oxygen gas. If you write ATP, what gives the ATP is the ADP and PI, because it's a cycle. If you see NADPH, the unloaded version is NADP plus H plus. Right? You don't have to count the number of ATP because this is not the cellular respiration. Okay, so this is it for the light dependent reaction. Let's do the light independent reaction. Don't try to remember these things, huh? No need. Whatever the side you put the ATP, the opposite needs to have ADP and PI. Whatever the side you have the NADPH, the opposite side needs to have NADP plus H plus. Okay, so the next one is the light independent reaction. So for light independent reaction, you're going to use carbon dioxide, the remaining one, and build up into glucose, right? So you're using carbon dioxide building up into glucose in light independent reaction. right? So light independent reaction, it does not need any light, so it's take place inside the stroma. What happened in the light independent is carbon dioxide building up into glucose. But this is an anabolic process. You're going from 1 carbon dioxide into 6 carbon glucose, right? So carbon dioxide has only 1 carbon atom. I represent it using 1 circle, but there are 6 carbons in glucose. So we have to build up into a big molecule. So always the building up reactions. So if I ask the most important reaction out of light independent and light dependent, if you are a plant, it is the glucose production, right? So glucose production happens in the light independent. Because plants doing the photosynthesis to get the glucose out. Because in cellular respiration, they can break it down to make the ATP. So the main reaction in photosynthesis is the light independent, but plants can't just do the light independent because some of the products need to be present